Okay, so good evening. I'll just share my presentation as well. Let me know if my presentation is visible to you all. Like basically the screen is shared. Yes, you can show thumbs up as well, so I don't have to look at the chat box. Yeah, great. Okay, I hope you all can see the presentation too. Yes, everyone. I'll add this as well. Okay, so this is an NPTEL course which has a code of BT57. And this course basically is upon gel-based proteomics and a bit of proteins as an introduction. Majorly, the course will deal with proteomics part and doing proteomics using a gel. Like there are different ways of doing proteomics, so we will be learning only gel-based proteomics in this specific course. So, giving you a bit of details about this course. So, this course is actually taken by Professor Sanjeeva Srivastava from IIT Bombay. Similar, myself, I'm Gaurav Bingi, and even I'm from IIT Bombay, currently pursuing PhD from Chemical Engineering Department under Professor Pramod Wankikar. So I've been selected as a TA for this specific course, and I've already taken this course even last year as well. And I'm taking this course again this year. So, to give an overview about NPTEL courses, so generally those specific courses are taken by one of the professors. We as TA, what we do is we give you like kind of help you all to revise the whole week's lecture as well as try and solve certain doubts which you all might be having while you go through those lectures. So basically, your aim is to go through those lectures. So you people might have been already having access to the week one lectures, all the five parts of those week one lectures. I think you all have those access to look after that. Yes, everyone. Have you all gone through that? Anybody? How many of you all have gone through those lectures? Like, if not in detail, at least looked after a few of those, or else some might have gone in detail as well. Can you all write in the chat box? I can see. I cannot understand this means whether you have gone through it, just gone through it, or gone through it in detail, looked all five lectures, or just two or three lectures. Can you all write it in the chat box or unmute yourselves and answer so that I know how much you have already gone through it, so that accordingly I can go in much more uh, detailed way or just give a background of all those parts, like all those lectures I meant to say. Okay, Manoj has gone through it, all the lectures. What about others? Okay, most of you have all have, I think, gone through the lectures. Yes, I'll be briefing it in like as much as possible. So what I do generally is like you all can suggest me in what way you all want the lecture to be. But this is my first lecture. Like after the lecture, I'll ask you all like whether this pattern of giving lecture is fine with you all, or if you all want to change the pattern, etc. We'll discuss about that. So I think few of you have all gone through those lectures. Some of you might have not. So I'll be giving you an overview of it. So basically, this doubt solving lectures, as I said, we as TA try and solve certain doubts and also give a brief revision of what sir has taught you all in those lectures. So it will be every Monday from 7 p.m. to 9 p.m. So if you any of your friends want to enroll in this specific course, today is the last day for enrolling, and the exam will be on 19 September for the same. So what we'll be learning in this specific course. So this is the first lecture today, week one lectures. Today is a basic lecture just about amino acids and proteins. We'll not be going much more in detail about what proteomics is or what gel-based proteomics is all about. We'll initially give a brief introduction of what exactly protein is made up of, how do we study proteins, what are types of proteins, etc. All this we'll be doing in this lecture. And then in week two, in the coming week, similarly on Monday 7 to 9, we'll talk about gel-based proteomics, give an introduction of it and how exactly samples are prepared for the same. Similarly, then after that, we'll actually learn about those different techniques which are used for proteomics analysis. Proteomics analysis. One is two-dimensional gel electrophoresis, and as well as in the next week we'll be doing about dyes, which is difference gel electrophoresis, and as well as a bit of mass spectrometry. So all of these will be done in this specific course. Today we'll be talking about only a bit of introduction part of amino acids and proteins. So in that week one lecture, today we will be talking about all of these, like introducing what are amino acids. So proteins are basically made up of amino acid. You all might be knowing it. So we'll talk about that a bit, introducing proteins, like the primary, secondary, tertiary structure, and all those, how exactly proteins can be identified, etc. Then about importance of folding to make a protein functional, misfolding about it, different theories which have been come up for understanding whether protein is folded or misfolded, etc. Talking about a bit of protein purification techniques, and as well as a bit of introducing what proteomics is all about. So before going into the lecture, I actually wanted to know uh, like a background of what you all are currently doing. Like, are you all a part of biological background only? Because in NPTEL, people also come from different departments. So if someone is from different field itself, I need to go a bit in detail. But so you all can let me know what you all are currently doing and basically your background. You can write it in the chat box like I'm currently doing this specific like BTech in this college or BSc in this college in biotechnology or in electrical engineering, whatever you all might be doing. Can you give me a background of you all so that I can accordingly go through my lectures? Okay, Manoj is already doing PhD. Like, is this your first year, second year, or just entered? Third year, great. Others, BTech biotechnology. I'm currently studying BSc in microbiology. Okay, BTech in biotech, MSc in biotech. Okay, Harshini is the junior most person here doing BSc in first biotech first year. Okay, BSc third year, BSc biotech first year. Akshata is also in her first year. BSc medical, B Farm fourth year. Okay, great. So most of you all are from a biotech background itself, biotech or basically biology background itself. So I don't think I need to give up much more details about current like today's lecture basically because today's lecture is all about introducing what are proteins and how exactly we deal with proteins. Okay, thank you for replying everyone. One is doing PhD in forensic toxicology, great. Alpana. Okay, some of you are doing B Farm as well. I don't know whether B Farm people might be having a like they are I think good in terms of chemistry compared to that of biology if I'm not wrong. Still, okay. So what I'll be doing is in the initial hour, so it's 7:15 now. So maybe approximately in an hour or so, all those five lectures in week one, whatever sir has uh, uploaded in terms of those lectures, whatever sir has taught you all, I'll just give you a brief overview of all those five lectures first, and then in the initial hour, basically in the next hour. we'll discuss few questions, and if you all are having any kind of doubts, we will try and solve those. And after that, also I'll try and ask you whether this pattern of giving lectures is fine with you all, or should I come up with more number of questions and giving lesser time for briefing the lectures, etc. We'll talk about that after my explanation of the whole 5 lectures. Okay, can we move ahead, everyone? Yes, people. Okay, thank you. Okay, so as I said, I'll be giving you a brief overview of all this introduction, all 5 lectures. So these are all 5 lectures of week one itself. Let's start. So you all might be knowing a basic structure of amino acid, which is one has carboxyl group and amine group, and this is the variable group by which there are 20 amino acids which have been come up. So these are the 20 amino acids. So this side chain is the one which changes, and due to which there are different names like glycine, alanine, serine, etc. So if you see, these are the side chains. This is I mean carboxyl terminal, amino terminal, and these are side chains you all can see above, which changes for each amino acid. Proline is the only amino acid which has a closed structure compared to others, and fewer aromatic amino acids as well, fewer aliphatic amino acids, fewer polar in nature, fewer non-polar in nature, etc. That's the way we can classify amino acids. So this is how we can classify polar or non-polar, out of which in polar some are charged, some are neutral, some in terms of non-polar, some are aliphatic, aromatic, sulfur-containing, etc. So this is depending upon the side chain. They have been classified into different groups. If they have a negative charge, then negative charge, positive charge. If they don't have any charge, we can call them non-polar, depending upon that. So that's how this is one of the way of classifying amino acid. There are different ways to classify. In amino acid itself, there are some L isomer and there are some D isomer, depending upon the polarized light being. So if there is an amino acid present in between the polarized light, if the light is going towards left side, it's called as L isomer. If right side, it's called as D isomer. So these are basically amino acids are non-superimposable, indicating they are mirror images of each other. They cannot be superimposed on each other. If you superimpose all of these together, R will go here and H. So basically you cannot superimpose them. So that's why they are called as non-superimposable. As the side chain is the one which changes. As I just mentioned about optical properties, about L and D. L isomer and D isomer, and those two mirror images can also be called as enantiomers basically. So this is what I was talking about. Left is levorotatory and right is D1 is dextrorotatory. So it's basically rotate the polarized light either to left or right. Depending upon that, there can be L isomer or a D isomer. And when you analyze proteins, by using a UV spectrophotometer, generally at 280 nanometers, approximately 280 nanometers. So these are the specific lambda max of each and every amino acid here. So generally, if a whole, so as you all might be knowing, amino acids, proteins are made up of amino acids. So if proteins have high amount of tryptophan, tyrosine, or phenylalanine, then only those type of proteins can be analyzed through UV spectrophotometer, as because the presence of those specific amino acids in the protein gives maximum absorbance at that specific wavelength basically, and that's how you can determine whether your sample has protein or not in it. So this technique will not be applicable if your protein doesn't have tryptophan and tyrosine in high amount. If there is only one or two, the peak might not be observed in 280 nanometers. So this is only applicable for those proteins which have tryptophan and tyrosine in it. This is another way of classifying amino acids called as essential or non-essential. So the name itself says that essential meaning we need that, it is not it, and it might be should be included in our diet, and it's not produced by our body. And non-essential are the one which are produced by our body. Then this is about isoelectric point, if you all might be knowing. So every protein has a specific charge depending upon the pH. So at specific pH, it has specific charge depending upon the type of protein it is. And a point at which it has no charge or zero charge, that specific point is called as isoelectric point. So that changes depending upon the pH for each and every amino acid. So this is how the changes is depending upon so acidic condition or basic condition. Accordingly, H plus ions will be added or removed, etc. So depending upon that, there can be either negative charge only, it can be only positive charge if there are higher H plus ions present, and there can be two charges together plus and minus indicating zero charge, which is a deuterionic form. So depending upon the pH, depending upon the amount of H plus ions available in the solution, accordingly, a charge of an amino acid changes. And basically, to what is a dipeptide? If you want to join two amino acid, there is a peptide bond which is being uh, formed. So dipeptide is nothing but two amino acids together, and the bond at which the connection is called as peptide bond. And there can be cis form of proteins are transformed depending upon the side chains are at a same location or opposite location. So accordingly, cis form of amino acid or transform of amino acid exists. Then proof for relation between amino acid sequence and in conformation. So basically, amino acid sequence is called as primary structure of a protein, and depending upon the sequence, a secondary structure is being like formed. Basically, if the sequence is different, the secondary structure will be different. So if you all might have heard about alpha helix structure, beta pleated structure, etc., that whole type of structure it will be forming depends upon the sequence. So there are different experiments people have done in order to prove that. And since an experiment which is about denaturation and refolding, so people have used different chemical to denature it to break the disulfide bond, etc. So these chemical like mercaptoethanol is used for breaking disulfide bond. This is used for denaturation of the protein. Similarly, ribonuclease A. So urea and guanidine chloride, etc. This effectively disrupt the non-covalent bonds of proteins, which are not strongly bonded. And when you add those specific, and whereas mercaptoethanol is the one which can cleave the disulfide bonds present in the protein. So if there is a protein, so there are two amino acids which have sulfate in it. Can you all know? Can you all tell me which are those two amino acids which has the sulfur group in it? Yes, people. Are you all listening to me? So there's cysteine and methionine. Yes, great. Suparna, Tuhin and Veena. Cysteine is for sure. There's methionine as well, which has sulfate in it. So basically, if in an amino acid sequence, if they are present, there is a possibility of forming disulfide bond. And if you want to denature that, you need to use one of the chemical like beta mercaptoethanol. There are other types of chemicals as well, which helps in breaking disulfide bonds. So one is beta mercaptoethanol. So those can be broken down and proteins can be denatured. Similarly, so this is an visually seeing how it like urea and beta mercaptoethanol can change the structure of a protein. For example, this is a folded protein, and you want to denature that, then this specific chemicals can be used, and all these are so as we learned, these are the one which breaks the non-covalent bonds, and mercaptoethanol is the one which breaks the disulfide bonds. So whatever they are, they will be broken down and it will be linear in structure, and they can be reformed as well. So this is the same thing, breaking up disulfide bonds here and non-covalent bonds here, urea and beta mercaptoethanol. And so when you remove the urea and beta mercaptoethanol, so they will be reformed again. So if there are traces of beta mercaptoethanol, so basically scrambled ribonuclease and so this is the protein they are targeting. So this is the scrambled one, which is not exactly formed because this 95 is need to be bonded with 40, and so basically these two are not exactly bonded together properly. Scrambled protein is something but which has wrong pairing, and due to presence of trace amounts of beta mercaptoethanol, so they are rearranged, and that's how the pairing can happen. So basically, denaturation and renaturation can take place with the help of these chemicals. And once that is done, if you want to reform it again, you need to remove it by dialysis. Then that denatured one can be reformed if those chemicals are not present in the solution. And once it is reformed, it can again gets its activity as well. So enzymes refolding can also take place, and it can be active also. That's about amino acids. That is what Sir talked about in his first lecture about introducing amino acids. Then in his second lecture of week one, Sir talked about giving an introduction of proteins where we talked about different structures. So that amino acids, if we join them together in a linear pattern, that's called as primary structure of a protein. Then depending upon those bonds being formed, it can form either alpha helical structure or beta plated structure. So we'll talk about all of those different levels of protein structure. So what is a protein? It came from a Greek word proteos, and it is a linear polymer built of monomers basically. Basically, the monomers are nothing but amino acids. And this is the most versatile macromolecule present in the living system. And it has different roles. So all these roles are being played by protein itself, like signaling, transporting, catalysis, helping in making the structure of a cell, etc. Everything. All those, there are n number of different functions of a protein. So these are certain examples of those functions, like transport. Hemoglobin is the one which transfers oxygen in our body. Insulin is the one which gives message to the cell to use the glucose. Then there are structural support like keratin, which help in making the structure of different parts of our body. In the muscle itself, there is some actin protein which helps in movement. There are enzymes like DNA polymerase which helps in replication of DNA. There is defense proteins like such as antibodies which helps in immunology or defending the foreign bodies present in our body, etc. Out of which most abundant protein is ribose coenzyme. So the bond between two amino acids is called as peptide bond, and this linear structure is the primary structure of the protein. There is carboxyl group at one terminal and amino group at one terminal. So these are secondary structure, which is alpha helical structure or beta plated structure. So they don't form randomly. There is a way of forming beta plated and alpha helical structures. So we'll talk about that how exactly this alpha helical and beta plated structures can be formed. So this is about alpha helix. So this can be formed between the first carboxyl residue with the fourth amino hydrogen of the fourth amino acid. So this is the first amino acid and fourth amino acid, second and fifth one. So they bond together in order to make a alpha helical structure. And if you see this, because of those attraction between the first and the fourth amino acid, and they make a turn, and that specific turn happens at a angle of 3.6. So there are 3.6 amino acid residues turn per helix, and this is the phi and psi angle which is being made with the help of alpha helical structures. And in terms of beta sheets, which is another common secondary structure which is generally seen in terms of proteins. But there are two types of beta sheets, either parallel or anti-parallel. If they are going in a similar direction, those two amino acid sequence when the structure is being formed, then it is called as parallel. If they are in opposite direction, it's called as anti-parallel. So if you all can see, this sequence is going in a same direction. If you see here, that's called as parallel beta sheets. If they are going in anti-parallel form, that's called as anti-parallel beta sheets. And they are also found in one specific pattern, They are not formed in a randomly because these are the only angles, phi and psi angle, which are feasible for making a proper protein structure. So if you see this, these are going in anti-parallel opposite direction, which makes them anti-parallel beta sheets. Then these are called as turns and loops, which you all can see there where there is no specific parallelity or anti-parallelity. This specific part of the protein structure is called as turns and loops. So then loops are rigid generally, and they are on surface. So we can differentiate between them how those angles are made. These are rod-like structures. These are sheet-like structures. Then there is H bond between amino terminal and carboxyl group in the same polypeptide chain as we just observed. Whereas in terms of this, there is H bond between amino and carboxyl group in different polypeptide chains. These are certain examples which have alpha helical structures. Then these are certain example of protein which have beta sheets. What is a phi and psi angle? So phi angle is between amino group and the C alpha carbon, whereas the psi angle is between the C alpha carbon and the carboxyl group. So depending upon this specific angle which is being formed, that makes so those angles are possible. So if you all might have heard about, have you all seen this specific graph anytime? Most of you all might have. So this is Raman's. Uh, which Raman was the one who gave these specific angles by which, yes, anyone wants to say something? Yes, was anyone trying to talk or speak something? Okay, so he was the one who gave this specific angles. So this is psi angles here, and these are phi angles. So the red part is the one wherein the structures are most comfortable in order to be stable in terms of a protein. So these are angles, phi and psi angles. So these are plus 90 and minus 90 about phi and psi angles, which make anti-parallel and uh, parallel beta sheets. And these specific angles are suitable for right-handed alpha helix, and these angles are suitable for left-handed alpha helix. Whereas all these angles, which are not being shown in yellow or red, they are not suitable for a protein structure to be stable. Then that was about secondary structure, and all these angles are about either alpha helical structure or beta plated structure. So that's about one specific sequence and how those primary and secondary, alpha helical and beta plated structures are being formed. In terms of tertiary, there are few more bonds which are applicable in terms of make a complete 3D structure of a protein. So this is the so like disulfide bonds, etc. There are different interactions like van der Waals forces, hydrogen bonds, etc. All those come into picture when a complete 3D structure needs to be formed for a protein. And quaternary structure is the one which requires different polypeptides together. So there are some proteins which require two, three, or four polypeptide chains together. If they so the bond which is found between two polypeptides or three or four polypeptides together, that is required in order to make a complete functional protein, and that specific structure is called as quaternary structure of a protein. So this is an example of quaternary structure of a protein which requires four specific polypeptides. So this is alpha 1, beta 1, alpha 2, and beta 2. So, and in terms of fetal hemoglobin and adult hemoglobin, there is some difference depending upon the type of alpha, beta, or delta or gamma chain is present in terms of. So, in terms of fetal hemoglobin, we require alpha as well as gamma hemoglobin in order to make a complete fetal hemoglobin. And this is the structure of how hemoglobin looks like. And so in order to detect any protein structure, crystallography is one of the technique by which we can determine the structure of a protein. So you need to purify a protein, crystallize that specific protein. There are different protocols to make it. Once you have a crystallized protein in your hand, you need to place it between an X-ray beam. Then when the protein goes through that X-ray beam, it makes a different pattern because of diffraction of X-ray, and using that pattern, people can determine a specific structure of a protein. That's how protein structures are being predicted till now. One of the ways X-ray diffraction. Then this is about oxygen binding curve, how exactly oxygen binds with, as we talked about, hemoglobin and myoglobin are the one which transports oxygen, but the way they carry it, depending upon the fractional saturation. So this is the graph of which by which how does a protein carries oxygen. Then this is an overview of whole second week lecture. Primary structure is the sequence of amino acids. Secondary is the about local folding within the chain itself. Tertiary is overall folding. As I said, there are a number of different bonds which come into picture for making a complete 3D structure. And if there are of different subunits like different polypeptides together, interaction among them makes the quaternary structure of a protein. Then there are different diseases as well for a specific protein. Like this is sickle cell anemia deals with hemoglobin protein. So only one specific change in the uh, nucleotide which makes some other protein itself, because of which this is generally how RBC looks like. But due to change in one of the specific amino acid, changes the protein. So if you all know how proteins are coded, if you all know about codons, do you all know about codons? Anyone here? Yes, you might have studied about that in your 11th, 12th standard, or in your first year you might be studying, like the one who are doing BSc here. Yes, you all might have heard about codons, right? How a protein is coded. Yes, no? I'm asking those BSc first year students, like the one who is in third, fourth year. Uh, they might be knowing, of course, but or in MSc. But I'm specifically asking BSc first year students who might or might not be knowing it exactly. Or so basically, codons are the one which includes three nucleotide sequence which codes for one amino acid. So even if there is a change in one specific nucleotide, it can change the protein. It can change the amino acid basically. So there is a change here, and there is a change in one amino acid which has led to this specific disease. So change in one nucleotide can change the type of disease basically. So here, this is hemoglobin, this is sickle cell mutant one. There is a change in A to T here, which made glutamine to valine. So basically, amino acid in the introduction part, I might have missed that. So amino acids can be coded in a three-letter code or a one-letter code. So this Lu indicates leucine, Pro indicates proline, Glu indicates glutamine, SER indicates serine, etc. So amino acids have different codes. It can be either three-letter code or there are one-letter codes as well for uh, identifying proteins. So similarly, like sickle cell anemia, there's one more disease which is beta thalassemia. So that if there is a change in this part of the sequence, so this is the normal thalassemia gene which is generally present. But if a person is suffering from beta thalassemia, there's a splice site here which makes causes that specific disease. Similarly, because of the change in few sequences here and there, people deal with Parkinson's as well as Alzheimer's disease. So allylysine, so this specific lysines come together and aldol is being made. So this specific group here makes aldol, and due to which lathrism is also caused. So these are basically different diseases due to change in one specific amino acid or the way by which those specific amino acids are interacting with each other. Different diseases can be caused. So this is about protein folding. That was about lecture two in terms of introduction of proteins. Then sir came to protein folding. So this is one amino acid sequence. This is another sequence, and this is how the proteins got folded. So what makes them this folding different? That we'll talk about in this lecture. So how does a protein exactly folds? It depends upon how is the sequence, and depending upon the sequence, are there any polar amino acids in the sequence, non-polar amino acids in the sequence, etc. So generally, how polar amino acid will do is all those polar amino acids present in the sequence, they might be present anywhere in the whole sequence. There might be, for example, it is an 100 amino acid sequence. In that 100 amino acid, maybe the amino acid number 2, 5, 15, 23, 25, 30, 32, all those middle amino acid might be of polar in nature, for example. So what they do is they tend to gather on the outside of the protein generally because they are the one which help in dissolving the protein because they will be having certain charge. They can interact with the solution and help in dissolving the protein. Whereas non-polar, they generally doesn't go outside. Non-polar are the one which are generally buried inside. So depending upon the type of sequence, that's how the protein starts getting folded. So this is what I was talking about, abbreviations of amino acid. It can be the three-letter code or one-letter code. And as I said, so these are the one which have these are polar in nature, having certain charge. There are certain non-polar amino acids. There are some neutral amino acids as well. So this is an example of the same thing which I just talked about. So for example, these brown ones are polar amino acids. They tend to go outside, and the green ones which are non-polar, they tend to be inside the. So that's how the folding takes place. This is the same thing which we just talked about. If you add urea, etc., they can be denatured, and if you remove urea, they can be reformed again. This is just the experiment by which people have done denaturation and then removing them, then again it got refolded. So for a specific amino acid sequence, there are n number of unstable conformations. There might be only one stable conformation through which protein can be functional in nature. So that specific structure a protein needs to take, like takes its shape to take its shape, and then only it will be functional in nature. So all those angles which we were talking about. Just a minute, I'm coming back. Yes. So we were talking about protein folding. So for a protein, there are depending upon the sequence. So there are different ways by which protein can get folded. Out of which there will be only one way by which it will be functional, and it will give you the stable conformation. So folding is a cooperative process. In general, any protein adopts only one conformation, which we just talked about, and very few closely related characteristic functional conformations is the native state of the protein. So this is just an example of the same thing. This is the folded protein. When you add the denaturant, increase the uh, denaturation concentration, it will form the unfolded protein. So this is the unfolded one. This is partially folded one. So all this can be done with the help of uh, like this experiment can be done with the help of urea, beta mercaptan, ethanol, etc. And people can understand how protein folding is taking place. So if you might have studied about free energy, entropy, and enthalpy. So if a protein is unfolded, it is so the amount of free energy. So this is the percent residues in native conformation, which is going down, and that's how the entropy is decreasing. And depending upon the native alpha helical structure, the entropy will obviously change because it will not be stable. So entropy will be lesser only when the protein is completely folded and stable. What is entropy? Is major degree of randomness, right? So depending upon that, a protein, like if you check their entropy and enthalpy, sorry, no, just the entropy, then determine it's depending upon the structure of a protein, whether it is folded or not properly folded. So beginning of the helix formation, the entropy is much more greater. So there is high free energy and helix formation commences and brings the energy, then entropy decreases. That's what I was just talking about. Then this was an experiment where people talked about two proteins which help in protein folding, that is DNA, J, and so not proteins, but maybe we can call it as enzymes, which helps for uh, which is also called as GROW-EL system, which helps in protein to get folded properly. So this is the cycle by which a folded native conformation can take. For example, initial, if there is an unfolded protein, DNA-J and DNA-K protein acts with it, and along with it needs certain energy so that protein is getting folded. The two phosphate ions are given away, and the folding goes on properly. Then there is one more group E enzyme which takes part in the reaction. And with the help of all of these specific enzymes here, a protein gets its properly folded structure. So, Sir has given an animated form of video. Like, if you all go through these lectures, you all can uh, take a look about how, like, in a Sir tried to explain in an animated video how protein folding can take place with the help of these specific enzymes. So that's the same thing, like using these two enzymes, DNAJ, DNAK, are partially floated. So this is an enzyme which gets regenerated for next round. So once you like ATP will go away, and again those specific proteins can come in contact with some other unfolded protein and make it folded. And talking about misfolding, so if there is a newly synthesized protein, it needs to be correctly folded without help or correctly folded with help of molecular chaperones. So chaperones are the one which helps in. So there are different chaperones present in our body as well, which helps the protein to get folded properly, because protein to get folded on its own is a bit difficult task, as we just talked about it. Like a protein can take a number of different shapes, but only one shape is the stable one and it will be functional. So for achieving that, there are different chaperones are also available, which helps the protein to get properly folded. And if it is an incompletely folded, so it is generally digested by the proteasome, because it will be any which ways it will be non-functional in nature. So if it is unable to take a properly folded structure and being make it functional, if it is not happening, the proteosomes are present in our body which will digest that specific protein. If that is also not happening, then proteins are generally get aggregated and they form insoluble fractions, because protein is not completely folded properly. Polar amino acid are not outside, like facing the surface in order to get dissolved. If that is not happening, then proteins generally get aggregated. So that accumulation of misfolded proteins, what happens to them? So if it is incomplete protein, proteasome are the one which digest it. So what happens to this? Where am I? So those misfolded proteins are the one which will be digested by proteolytic enzymes. So proteolytic fragments result into few. So those specific part can also, like, because of the degradation by the proteolytic enzyme, they can lead to few degenerative diseases as well. So that is characterized by the presence of insoluble proteins or plaques present in brain or liver cells. So that depends upon the type of protein basically, and these are certain types of diseases which can be caused because of the presence of some type of proteins, and if they are insoluble, and they can be determined, uh, like if they are present in such body parts, and those are called as degenerative diseases basically, and these are those diseases. So that was about part three, or third lecture of week one. Then, in terms of fourth lecture, so I talked about purification techniques. So now, for example, if you made the protein, you have a protein, but all those are made inside a cell, right? And you need to bring them out, or have a purified protein outside the cell. So you all can do it by lysing the cell first. Then you have a lysate with you, and that lysate can be purified with the help of different techniques. One of the technique is chromatographic separation. So this is a column protein separation wherein there is a solid porous material, there is a mobile phase, and when you add your sample, depending upon the affinity between the solid phase and the mobile phase, like the stationary and the mobile phase, depending upon its affinity, it will depending upon the type of mobile phase you are using. Accordingly, those proteins can be separated, and you can get a purified protein out of it. So there are different types of uh, protein purification techniques. Some are like gel filtration, ion exchange chromatography, affinity chromatography, HPLC, etc. So this is one of the techniques. So what are you doing in this chromatographic technique? Basically, you are, as I just mentioned, depending upon the differential solubility of proteins. So these are different principles by which a protein gets separated, either depending upon the solubility, depending upon size, depending upon charge, or depending upon affinity. So chromatography involves four major components: mobile phase, stationary phase, detector, and sample introduction, etc. And it requires a solid material, which is a matrix, which is can be beads, can be which is basically beads, and those beads, depending upon the size, depending upon the porosity, and depending upon the charge, there are different types of purification technique. So as I just said, matrix is generally beads, and they may or may not be attached to a chemical group depending upon the type of technique you want to use for purifying the protein. So in terms of gel filtration, it can be also called a size exclusion chromatography. So here the separation depends upon the size of the protein. Smaller the size, it will be it will be moving through the gel matrix and it will elute out later, whereas larger size protein will elute out first. So this is the visual representation of the same in terms of size exclusion or gel filtration chromatography. So the larger protein molecules are the one which will not enter into the beads, and that's the reason it will get eluted out faster. Whereas there is a longer path for smaller proteins which will enter the beads, and accordingly slowly they will come out. So they elute out later. So initially you will get larger proteins first in initial fractions and smaller proteins in the later fractions. And all those fractions can be analyzed through UV spectrophotometer as we just learned about it. You can measure it into 80 nanometer, or else there are different protein assay kit, protein assay, uh, what do we say? There are different assays as well to determine proteins such as, okay, I can ask you a question. What are the different types of assays you all know for determining a protein? If you all know any of it? Yes. You all know how proteins can be detected? Have you all heard about any assay which helps in determining the protein? Yes, people, no idea. Biuric acid, fallen Lowry. Okay, Lowry is fallen Lowry acid. Yeah, Bradford acid, etc. Great. So these are certain. So basically, after purification, you all can determine those fractions can be analyzed by a Bradford assay or just by UV spectrophotometer or foreign Lowry assay, etc. And you can determine what like if what is the concentration of protein present in it. So this is about UV spectrophotometer. Then depending upon the concentration, you can get the uh, protein concentration, and initial fractions will generally have higher molecular weight proteins, and later fractions will generally have lower molecular weight proteins. That's about size exclusion. Then there is method called as ion exchange chromatography, which deals depending upon the charge, a protein gets separated. So pH is the one which influences the net charge of the protein, which we talked about. So the buffers which you are using in terms of separating these proteins, uh, like the mobile phase you will be using, that should be accurate in terms of pH, so that the charge is not getting affected. So if you want to separate positively charged amino acid, positively charged protein, you can use cellulose, which is negatively charged. Sorry, you can use carboxymethyl, which is attached to a cellulose or agarose beads, as we were talking like it can be with or without chemical attached, right? So if you attach carboxymethyl to a cellulose, that helps in separating positively charged amino acids, positively charged proteins, and this helps in separating negatively charged proteins if you have diethyl amino ethyl DAE beads. So depending upon the need, depending upon the type of protein you want to separate, you can use these beads. So for example, if beads are negatively charged, the negatively charged uh, proteins will elute out first as there is no interaction between them. The positively charged protein will have some interaction with these beads because opposites attract, and because of that, those are eluted later. That's about ion exchange chromatography. In terms of affinity, there is a in the to the bead itself there is some affinity, such as nickel or something is attached to it, so that affinity comes into picture. So metal chelation is widely used in purification. There are certain substrates, products, cofactors, antibodies, etc. So basically, that is chemically coupled with any of the material, so that proteins are attracted to it. So here, the red one protein is basically attracted to this uh, entity which is attached to these beads. So, for example, his tags are generally attracted towards nickel. So histidine, so if your protein is having uh, histidine tag. So that is can be purified using affinity chromatography, wherein beads are with the nickel, and you can get those proteins eluted out, the protein of interest which has histidine tag, whereas all other proteins will be eluted out first, and you can get the desired protein using affinity chromatography. So these are different ligands fusion partners. As I was talking about, I gave this one example, histidine and nickel. So depending upon the type of protein fusion partner you are having, you can have different ligands being attached to these beads. Here, all those are available commercially. Accordingly, those beads can be taken uh, and used for elution of those specific proteins. And accordingly, those elution buffers are used for the type of protein you want to elute. Yeah. So it's the same thing. Then when you do it on SGS page, you will see those protein bands, purified ones. You can also use uh, automatic techniques like HPLC, etc., wherein mobile feeds are being provided. You also add your samples. Similar concept, but using high pressure liquid chromatography, and you can still separate the components. So there is you can adjust the flow as well, and depending upon the fractions being collected, similar way you can determine the proteins. So this is mobile phase A and B, HPLC pump. These are different buffers which can be used. So in terms of, so based upon the hydrophobic binding, on the, so as I just said, depending upon its interaction with the solid, which is RP column here, or the liquid, the mobile phase or the stationary phase, depending upon its interaction, the separation can take place. Then SCX resin, which is strong cation exchange. So these are nothing but silica based cation exchange. So this whole column, so for example, here it is reverse phase column. Similarly, this column will have such kind of ligands attached to it, which is silica-based cation exchange stationary phase in that column itself. So depending upon the protein, if it is sulfonic acid cation-based exchange ligand. So this is nothing but an example of ion exchange chromatography, wherein ligand covalently bonds to this polymer depending upon your protein, and those are separated, eluted out later, whereas the other proteins can elute out first. In terms of proteomics, so proteomics is nothing but a study of protein. If you are studying the gene, it's called as genome or a genomics. If you are studying the transcript part of it, the gene gets transcripted to RNA. So if you are dealing with transcripts, it is transcriptome, and if you are studying about that, it is transcriptomics. Then that transcript gets converted to protein. If you are studying about those proteins, it's a proteome, and if you are studying about that, that's called as proteomics. So steps involved in proteomics analysis is, so as we talked about, you need to, so all those are present inside the cell, right? So you need to extract that protein out first, then you need to separate that, purify it, and you have separated. Now you don't know in which fraction what type of protein is present. You need to identify that protein. That identification can be done with the help of mass spectrometry, or if you have standards, you can just compare it with the illusion peak, etc. And then you can characterize it, the same protein. So if you're doing all of these, basically you're understanding protein and trying to understand what protein is. That is nothing but proteomics. Everything about whatever you're doing, dealing with proteins, understanding proteins, that is nothing but proteomics. So this is the I have 28% chance. Okay, I'll just finish this up. I think there are a few more slides left, and then I'll keep the charging. Okay, so this is how the history of proteomics is basically. So there was 2D gel electrophoresis wherein proteins were separated with the help of electrophoresis technique. Then mass spec came into picture. Even uh, earlier itself, there was nucleotide sequencing also came up. There were mass spectrometry which came into picture in the year 1990s, in 1990s basically, and that's how proteomics gained its emergence because MS is the one which actually speeding up the proteomics process because that's how we are able to identify what protein we are separating or what we found in a much more accurate way. So in terms of proteomics can be divided into two, classified into two. Either you're talking about functionality of the protein or abundance. If there is abundance, you need to identify, quantify it, or functionally, if it is having certain expression, you can perform an assay as well. So these are the components of omics in a broad way. We just talked about if you're studying genome, it's genomics. Environmental effects is called as epigenomics. Transcriptomics, we are dealing with transcript. If you are dealing with proteins, it's called proteomics. If you are dealing with metabolites, it's called as metabolomics. So this is mass spec itself. We'll talk about mass spec in detail maybe in later lectures. So it is nothing but you are giving your samples here and mass spec, depending upon the mass, it separates mass to charge ratio basically, depending upon the charge as well as the mass. That is mass to charge ratio, whatever the ratio comes, depending upon that, these are getting separated and you get certain eluted peaks accordingly depending upon mass to charge ratio, and that's how a protein is identified. It's about alternative splicing. So this is DNA having all the exons. So there is exons and introns. Basically, exons are the one which are which are called as coding sequences, and introns are the one which are called as non-coding sequences. Those introns are removed, and exons are the one which come together to make a functional protein. So alternative splicing is required because one RNA can code for different proteins. So if 1, 3, and 5 exon come together, they make one type of protein. If 2, 3, 4. This is just an example how alternative splicing works. And that one mRNA can code for three different proteins as well, depending upon those exons coming together. Along with that, there is something called as post-translation modification as well. Once a protein is like RNA is getting translated to a protein, it can get modified by phosphorylation, glycosylation, methylation, hydroxylation, etc. So those are a part of post-translation modification. All those needs to be studied if you want to study proteomics. This is what we are talking about. Bradford assay is one of the way by which we can determine uh, if a protein is present in your sample. So how does this specific reaction happen? So there is CBB reagent present in the Bradford method, which is Comacy brilliant blue, and that is the one which interacts with the protein like this. And when they interact, they form. So basically, a bluish to purplish color is being formed because of this interaction, and that can be determined at a lambda max of 595 nanometer. So higher is this specific uh, lambda max being obtained can tell you about the amount of or the concentration of protein is being present. If there is no protein present, then it will show nothing because its lambda max is 465. And if you are checking at 595, that will be only gain its peak only when a protein is present. So it makes a complex basically, and that's how you can determine the concentration of protein being present in this one of the method, which is Bradford methods. Then this is another separation method, something called as electrophoresis. In SGS there is stacking gel, there is resolving gel, and when you add your protein initially gets stacked in one specific line, and then they get separated depending upon the size. And so SGS gives all the proteins negative charge, and once that negative charge is the one which helps them for separating. So, so porous nature is the one which will help them to separate it completely, depending upon the size of the protein. The smaller are the one which moves faster in the porous, and larger are the one which will move slower. This is we talked about isoelectric point, at point at which it has no charge, so it will stop moving. If it has certain charge, then accordingly it will go above or below depending upon the charge. Then 2D gel electrophoresis, initially you will run isoelectric focusing experiment, then you will use that isoelectric focusing strip to do gel electrophoresis, normal gel electrophoresis, which is SGS page. Then we'll talk about DIGE as well in the coming lectures. So DIGE is differential gel electrophoresis. So okay, so this is 2D gel electrophoresis itself, wherein your samples will be labeled with different dyes and control as well as the treated one, and you can mix all of this together and label it with different dye and mix all of these, run a isoelectric focusing experiment with IPG strip and use those specific separated proteins and run them on SGS gel. And when you are visualizing this protein, you need to visualize depending upon the dye. So those will be visualized at a specific wavelength. And when you are analyzing those images, so this pooled one will have both the proteins together, whereas then you need to compare basically. So for example, this specific protein is coming from the red one, which is the control sample, which is present in disease sample also, whereas if you see about this protein here, it is not coming from the control sample, but is only coming from the treated sample. So that protein is specifically might be the one which is responsible for the disease, etc. So that's how you can determine using the help of 2D gel electrophoresis. So that is the five lectures which Sir talked about. So as I said, I generally give a whole quick review of whole five lectures in an hour. So I try to complete within in an hour. I'll just keep it for charging just a minute. Then we can discuss more on it. Okay. Okay. So that was a quick overview of what Sir talked about in week one lecture. I hope you all got the revision of it. Yes, everyone? Okay. So there is Manoj Jeba. Jeba, I hope I'm pronouncing it correct. There is Veena, Suparna, and Shivanjali, and Suzanne, and Himani, and there are two others, Alpana and is the other one, Pooja. Okay. So so we'll discuss few questions as well, like previous year assignment questions, etc. So I wanted to know whether uh, before that, like, do you all have any kind of doubts which you want to discuss, etc. So that, uh, like, or should I move on with the questions? Yes. You all want to discuss anything related to the lecture first, or should we, like, if you all have any kind of doubts which you all wanted to ask, etc., or should we move on with the question and answers session? Yes, everyone. Anything? Did you all get whatever I just went through the whole 5 lectures? Did it all help you all in terms of getting a quick revision of everything? I think only Veena is the one who to which I helped. Suzanne says yes, Himani says yes, Jiva says yes. Okay, super. Now. Okay, great. Uh, you can like it's okay if you all don't. You can all call me by my name as well when I'm a PhD student. It's just that I'm doing the TA teaching assistantship for this specific course because I'm from this field itself. So like even if you all can call me by my name as well. Okay, I think if you don't have any kind of doubts, so maybe we can move ahead with question and answer session, or do you all want a break of 5 minutes or so so that then we can start? If you all want to freshen up a bit, like go to washroom and stuff, you all can do that. Okay, should we start in 5 minutes or so then? Take a break of 5 minutes. Yes, everyone. Okay. Okay, then I'll just can you all see this questions? Yes, everyone. Okay, I'll just open chat box here so that you all can answer the questions and I can see it. Okay, so which are the functional groups that are common to all standard amino acids? You can tell me the option, or you can read the, you can just say A, B, C, or D, whichever you feel like, which you feel that's the correct answer. Okay, everyone is like A. Yeah, A is the correct answer. So as we, that's the first slide of the presentation itself. Like it has an amino group and a carboxyl group. So which amino acid is optically inactive? So this is an amino acid which will have so so alpha carbon is the middlemost carbon. Once there is amino group, one there is carboxyl group. And one side there is so carbon has four chains, right? It has one hydrogen and there is one side chain. And that side chain needs to be similar thing of similar molecule as that of either amino group, carboxyl group, or hydrogen, and that will make it optically inactive. If all four are different, it will be optically active. So, as you most of you all know the answer, I think. So, I hope you all understood. The people who don't know why A is the answer, I just explained a bit about it. So glycine has hydrogen itself in its side chain, and because of that, that hydrogen and other hydrogen. So there are two same molecules in with the carbon attached to it, whereas one there is amino group and carboxyl group, which makes it optically inactive. If all four are different, then it makes optically active. What happened? Okay. So the chirality of amino acid is due to what? Yeah, it's due to, as I just mentioned, it's because of asymmetric alpha carbon. Okay, the primary structure of a protein refers to. Okay, yes, it's the sequence of amino acid. So can you all tell about 3D conformation in the tertiary structure? What is the C option? What type of a structure it is? Is it primary, secondary, tertiary, quaternary? It's a part of what? So 3D obviously is tertiary. Like I don't have to ask that question because of the presence of 3D there. What is assembly of subunits? Does it come into picture in secondary structure or primary itself or tertiary only, or is it about quaternary? Yes, yeah, it is about quaternary when your two subunits are attaching. Then hydrogen bonding pattern is for the D1 stands for what? Secondary. Yeah. So that's how alpha helix and beta pleated. There is a pattern by which hydrogen bonds are formed and you get the structure. So which bond is not involved in stabilizing protein tertiary structure? This is a bit tricky question though. Yes, fifth one. Yeah. So that's the primary structure, right? Peptide bond, making all amino acids together. That's whereas all others are involved in tertiary structure, out of which hydrogen is a part of secondary as well. So hemoglobin is an example of a protein with only primary structure, quaternary structure. So remember these options: no secondary structure or lipid components. So it's about a quaternary structure because it has four polypeptide chains. Seventh one, chaperone proteins. What does they do? Correct, protein folding. Yes. Who does protein degradation? Which what type of proteins? Yeah. What type of proteins do protein degradation? Like what enzymes are involved in protein degradation? Proteases or proteolytic enzymes. Okay, so in this we are not dealing with DNA and carbohydrate, so I'm leaving that. Alzheimer's disease is linked to misfolding of which protein? We have not read about it in the lecture, but maybe if you all know, you all can answer. I just said that there are different proteins because there's a change in some or the other, and that deals with different diseases. So if you all know this answer, maybe you all can answer, or else I can reveal it. Yeah, it's MLRP. So prion disease are caused by either by so this should be answerable, I think so. Is it because of viral infection, misfolded prion proteins, antibiotic resistance, or lipid accumulation? Protein is a helix breaker. Won't it be counted into a degradation of protein? It won't because proline has its own properties and it is, although it is uh, alpha helicase breaker because that is required so that protein takes its own folding wherever it is required and in order to make it function. Protein has its own rule. It is not considered as degrade. What do you mean by degradation of protein? It is completely broken off and you are confusing with denaturation of protein and degradation of protein. Like protein is still there. Like in denaturation, like maybe you are if there is a helix breaker, after that helix breaker, whatever is the other part of the sequence, it can take any other structure if it is needed. So all those is so proteins, so amino acids are placed at specific locations, and because of those specific location, protein is becoming functional. So if if a protein has proline in its sequence, it has some role in it. So it is not there just like that. As you all know, even if there is a change in one specific amino acid, protein can be non-functional also. So that is not considered as degradation. Yeah. So in terms of this, yeah, it is the B answer, misfolded prion proteins. So salting out in protein purification relies on what? Although we did do it in the lecture, but still. But all those are basic questions you all might be knowing. So we just talked about chromatographic technique, but along with that chromatography, one of the purification technique is salting and salting out as well. So that basically depends upon the salt concentration and the charge of the protein. So this is about solubility difference at high salt concentration. So which one does separate based upon size? A? Come on. It's B, it's FGS page. I mentioned the separation on the basis of charge. Affinity on the basis of affinity, basically. If it is getting has some affinity towards the ligand, it will accordingly separate out later. Whereas in isoelectric focusing, depending upon charge again, like depending upon the pH, the separation will be different. SDS is the one which separates on the basis of size. So Bradford assay measures protein concentration using what? Yes, it's B, it's Comasi Brilliant Blue. Proteomics is the study of what? Yeah. So 2D gelatophores, proteins are separated first by. So I just give an overview how 2D gelatophores work. There are two techniques. What is the first technique which is being used for 2D gelatophores, and what is the second technique? Yes, others also answer. I can see only two or three answers right now. Okay, so isoelectric point or isoelectric focusing basically separated first by isoelectric focusing, and second is SGS page, correct? So one of the applications for proteomics is So I think the options here are very easier to give a proteomics application. Maybe a more confusing option would have been a tricky one. So gene deals with DNA, RNA is for with the transcript. This is for metabolomics. This is metabolomics, transcriptomics, and genomics. So this is the only one which deals with. So this can deal with anything basically. This can go with any of the stuffs. Biomarker discovery, it can be either genomics, proteomics, metabolomics, anything. So biomarker discovery can be a part of anything. Okay. So these were the few questions which were a part of previous year questions. Hmm, okay. Still, so if you all have any kind of doubts related to today's lecture, you all can ask me. Or like I don't know. If, I just wanted to know if you all like this pattern of teaching. Like I'll go through the whole division in an hour or so, and then I'll go to the question and answers. Do you like are you all good with this specific pattern of discussing, or do you all want to change it, or do you all want to just deal with question and answer, or elaborate more on the brief discussion, like maybe 1.5 hours or so? Like how do you want it? So if you want more discussion, maybe I can uh, talk more about the lecture. Yes, you will be getting the PPT, everything. Okay, you want more questions related to it? Okay, I'll try and add that next time. I need to check whether I have a few questions. Okay. Yes, anything else you all want to suggest? So one suggestion is I need to add more questions. One if people wanted, what do we say, PPT of it? Yeah, and you will be getting PPT of the same. So no need to worry. I'll be uploading this recorded lecture as well. So you can go through the lecture again too. So there's no problem in it. So you can get the recorded lecture as well as the PPT. So can we get any hands-on training? Can we get brief discussion about BLI and SPR? What do you mean by that? Hands-on training at IIT Bombay. May you all you need to ask Sanjeeva sir for that. Like if he is willing to have one session with students who are interested to come to IIT Bombay. And what is BLI? So one is surface plasmon resonance. Okay, I'll consider that. And what is BLI? So Manoj, I think sir is taking a live lecture tomorrow, right? Or is it today? Was it happened today at 11 o'clock? Was it today or is it tomorrow? I don't remember. It's tomorrow, right? Okay, so Manoj, tomorrow at 11. So you can ask Sanjeeva sir itself, like if he is willing to give hands-on training. So if like where are you all from? Like if anybody else is also interested for having hands-on training. Yeah, from Haryana. Great. Yeah, you can ask sir. Like there is an email being sent to you all uh, regarding the live lecture session, so you all can find that. If you don't have that, maybe I can share it with you. You don't have that email, Manoj? Others, do you all have that email of Sanjeeva sir live lecture? So there is a link for Google Form as well. Like if you all want to ask any question to sir, you can ask. I think this is the live YouTube link. You can save it. People are from Kerala, Pune. So I've just shared it. So you can. So this will be the live session tomorrow at 11 by Professor Sanjeeva. So for hands-on training, you all can ask it. So if it is possible for sir, he can arrange one for you all. Yeah. So in my lab, I have SDS page and Western blot, but don't have isolatory focusing apparatus with me, neither for Dij. So just for SGS and Western blot, you all can come to my lab itself. If you are visiting IIT Bombay, I can uh, show you all that. But in terms of Dij, it's all those are available in Sanjeeva Sir's lab itself. So one of the students from his lab can train you all. So you can ask for it if there is a possibility. I don't know if there is a possibility or not, but if it is, maybe sir can arrange one for you all. Okay. Anything else? So there's a Google form as well. I think I can share uh, that link also. Just a minute. So in this form, you all can ask questions. So sir can go through those questions and accordingly answer in that live session. So you all can ask the questions prior as well, like prior to the live session. And during the session also, there will be a chat window as well. So you can ask during the session too. Right? Okay. So like there's one session of sharing PPT. It will be done obviously. Like I think you all might have given access to one of the Excel sheet wherein the link for the live session. This live session will be uploaded. So link YouTube link for this live session as well as PPTs which I'll be uploading in the Google Drive. So you will be given the links for the same. So you all can access that link and accordingly download the PPT as well as questions as well as uh, the recorded video. And regarding surface plasmon resonance and biolayer interferometry, I'll try and include about that in the like wherever it is suitable. Like I think during the third week lecture, I think that would be a best time to explain these techniques. So I'll add those things. Yeah, anything else you all want to ask or discuss? How is your like? I think just coursework might have started. Like along, have you all taken any other course as well in NPTEL along with this? Yes. No, I think only one course you all are doing. Okay, if you're planning to, I think the last date will be ending soon, so go through it faster so that you don't miss out. Even if you miss out in case, for example, you can actually email NPTEL coordinators and ask them if you want if you really want to join any of the course because TA sessions have been started like for week one for any course, so like people are already like started with it basically. Okay. So if you have any questions, do ask me. If you all don't have any other questions, you all can leave the lectures. If you have any doubts, you can we can still talk about it. We have 10 minutes. Okay, thank you. Thank you for attending the class. I hope I will see you all in the next lecture too. Okay, thank you. Thank you everyone.